Have you ever taken one of those DNA tests that promises to tell you where your ancestors came from? They give you pie charts, maps, and sometimes even a list of famous people you might be distantly related to. Well, underneath all of the marketing lies a deeper and more sobering question. What does our DNA tell us about where humanity came from, and how long have we been here? Thank you for joining me. In our last curious episode, we asked whether creation science is outdated. Today, we are going to look at a very specific corner of that debate: human genetics. And the strange fact that, according to modern genetics, all of us trace back to a surprisingly few ancestors. We are going to look at mainstream genetic research and the work of Dr. Nathaniel Jeanson, the author of Traced, and ask a simple question: does the genetic evidence really demand a very long human history, or could it actually fit a much shorter one? No pressure, no ultimatums, just an honest look at the data, models, and the tension between them. To follow this conversation, we need three key concepts: mitochondrial DNA, the Y chromosome, and MRCA, or most recent common ancestor. Mitochondrial DNA, or mtDNA, is a small loop of DNA inside mitochondria. You inherit your mtDNA from your mother, who inherited it from her mother. It traces a single, unbroken maternal line. The most recent woman from whom all living humans inherited their mtDNA is called mitochondrial Eve. This is a technical term for the convergence of maternal lineages. It does not mean she was the only woman alive at the time. On the other side, the Y chromosome is inherited from father to son and traces a single paternal line. The most recent man from whom all living males got their Y chromosome is called Y chromosomal Adam. Again, this is simply a convergence point. Both concepts rely heavily on mutation rates. If the rate changes, the dates change. This is where the debate truly begins. When scientists date mitochondrial Eve and Y chromosomal Adam, they are not dating the first humans. They are dating the most recent common ancestors for two specific inheritance lines: one maternal line, mtDNA, and one paternal line, Y-DNA. With that in mind, large data sets generally put both convergence points in the same broad neighborhood, on the order of 100,000 years ago, give or take. Different papers report different ranges, but the key point is that the ranges overlap. A major 2013 study by Posnick and colleagues used high coverage Y chromosome sequencing and a calibrated mutation rate and found a Y chromosome MRCA that overlaps with common estimates for mitochondrial Eve. That 2013 work is one reason you'll often hear that the two timelines are closer together than they used to be. Before the era of larger data sets and improved methods, the scientific picture was much less stable. Some early Y chromosome studies produced younger ages, some produced older ages, and the estimates moved around as methods improved and as more lineages were sampled. And then there was an attention grabbing detour. A rare, very deep branching Y chromosome lineage was reported in 2013 from a lineage connected to Western Cameroon. Using one set of assumptions, that analysis suggested a much older YMRCA on the order of hundreds of thousands of years. Later reanalysis argued that the extreme age was not required and that the YMRCA sits closer to the more typical range reported elsewhere. Why can the Y timeline swing like that? Because male lineages can vanish. If a paternal line leaves no surviving sons, that branch disappears from the living Y tree. As branches die out or new rare branches are discovered, the most recent common ancestor can shift. And through all of this, one thing stays true. The dates depend heavily on the mutation rate used to convert genetic differences into years. Change the rate, and the calendar dates move. Dr. Nathaniel Jeanson, who has a PhD from Harvard and works with answers in Genesis, argues that the mutation rates we observe directly in modern families should carry more weight than slower rates inferred over long time scales. In genetics, you'll sometimes hear this as pedigree rates versus phylogenic rates. Pedigree rates come from measuring new mutations from parent to child. Phylogenic rates are inferred by comparing lineages over much longer spans and are often calibrated against other timelines. Jason's claim is straightforward. If the faster, directly observed rates are the right rates to use, then the genetic clock runs much faster. If the clock runs faster, the same amount of genetic variation can fit in a shorter time. Using that approach, Jeanson concludes that human mitochondrial DNA diversity can fit within thousands of years rather than hundreds of thousands. And he argues that Y chromosome diversity fits within a similar short time frame following a severe bottleneck. It is important to say this clearly. Mainstream geneticists do not accept those young Earth conclusions, and they offer reasons for why short term rates and long term rates can differ. But Jeanson's work is not built on secret data. It is built on a different set of assumptions about which mutation rate should be used and how to interpret the same public data sets. So when we compare the models, we are not comparing data versus no data. We are comparing how the same data is translated into a timeline. Mitochondrial barcoding studies examine a specific mitochondrial gene called COI across huge numbers of animals. Scientists use COI because it is useful for identifying species. A well known 2018 analysis by Stokel and Thaler drew from a very large barcode database spanning roughly 100,000 animal species and millions of sequences. They reported a striking pattern. Within most animal species, mitochondrial variation is low. Humans fall right into that same general pattern. Mainstream researchers also recognize the low variation. Their interpretation is different. They commonly connect it to bottlenecks, founder effects, and population expansions in the late Pleistocene, not to a recent global flood. In other words, both sides can agree that bottlenecks and expansions shape genetic diversity. They disagree on the timescale and the historical cause. And this is a helpful moment to pause and be honest about what the barcode data can and cannot do. Barcodes can highlight patterns. They can show that diversity is often low within species, but a single mitochondrial gene cannot, by itself, settle the entire timeline of human history. It's one piece in a much bigger puzzle. Even though the timelines differ dramatically, both sides start from the same basic observations. Where both sides agree are all living humans share a common maternal lineage and a common paternal lineage. Bottlenecks and founder effects are real and they leave signatures in DNA. Human genetic variation is low compared to many other species. Humans expanded and dispersed quickly after major population constrictions. Where they diverge is number one, the time scale, where the mainstream says the key bottlenecks and expansions are placed tens of thousands of years ago, and the deeper lineages reach back on the order of 100,000 years. And young Earth creationists say the major bottleneck is placed within the last several thousand years with rapid post bottleneck repopulation. Two, mutation rate assumptions. The mainstream uses rates that best fit long term data sets and multiple calibration points, which generally yield older dates. Jensen's approach prioritizes directly measured present day pedigree rates, which compress the timeline. Three, historical framework. The mainstream interprets the genetic story within deep time and ties it to archaeological and fossil chronologies. While creationists interpret the genetic story within a biblical framework of creation, a catastrophic bottleneck, and a rapid spread of nations. The core contrast stays the same. The data points are often shared. The timeline depends on the assumptions used to interpret them. Where does all of this leave the curious? You do not need to settle the entire debate tonight, but you can start with a question that is hard to avoid. Why do multiple lines of genetic evidence keep pointing back to small beginnings and major population constrictions? Mainstream models explain this with ancient bottlenecks and long recoveries. Young Earth creationists explain it with a recent catastrophic bottleneck and rapid repopulation. If you're a skeptic, you don't have to accept a biblical conclusion to recognize that the timelines are not pulled straight from the DNA without assumptions. Mutation rates, calibration points, and model choices matter. And if you're coming at this from faith, you don't have to fear the genetic data. You can examine what it actually says, where the uncertainties live, and why intelligent people disagree on the interpretation. The Curious series is not here to push you toward a particular conclusion. We are here to walk beside you as you examine the evidence without pressure, without inherited assumption, and without fear. Because the Bible is not allergic to science, and science is not as settled or as one directional as many people assume. If nothing else, recognize how much information is written into our DNA and how that information keeps challenging our assumptions no matter which model we use. Stay curious, keep seeking, and don't be afraid to re examine the story you were handed.